Good afternoon to everybody. It's a pleasure to start this series of online seminars on multiscale phenomena in continuum mechanics, singular limits of equilibrium and transitions. These seminars are organized in the frame of the PRIN project coordinated by Marco Sammartino and involving researchers from Palermo, Catania, Messina, and Parma. Of course, it would be much better to organize and to attend in-presence conferences, but now the COVID situation is still unstable, and so we have organized this online series of meetings. We thank all the participants because we had a lot of registrations and we are very glad for this. It's an honor to have as first speaker of this series of seminar, the Professor Benoît Perthame from Sorbonne University, which is a renowned expert on the many topics related to the frame of our research project. In this talk, he will speak about mathematical models of living tissues and free boundary problems, about 40 minutes talk, and then we will have time for discussions. So please.
So you see my screen?
Yes, perfectly.
Okay, so thank you very much for the invitation to give this talk. It's a pleasure to be with you, even though it's online. So this talk will be about mathematics of living tissues and the connections to free boundary problems. The main motivation are problems issuing from cancer growth, tumor growth, and the numerical simulations about that. So to begin with, let me tell you that the modeling of living tissue is an ongoing problem, which is studied not only in mathematics, it's also studied very much in physics and mechanics. Just because we only begin to understand what are the process which explain how living tissue is behaving in terms of mechanics. So I've made a list of people with whom we have more or less connections in our team. They come either from physics or from mechanics or from mathematics. Basically what have been understood by all these people in the last 20 years, let's say, is that a tissue is mostly fluid, a complex fluid, and you see here how it is, how you can imagine it is made of. So you have this extracellular matrix, which you can understand as long polymers, which maintain the tissue. The most common of them is a collagen for the skin, for example, or for other tissues. So this makes the stiffness, and around that, you have different types of cells, which are moving through this extracellular matrix and using the extracellular matrix to move. These can be cancer cells or healthy cells, dead cells or living cells. Many kinds of cells can have different mechanical properties. In between that, there is a fluid, which you can think of as water. And in the water, you have molecules, which are the chemical species, which are even released by the cells and allow the cells to communicate and change their behavior. So because of that, it has been clear now that there is a notion of pressure in a tissue, a living tissue, which is not exactly the pressure you find in the usual fluids or gases. but it plays the same role in terms of fluid mechanics. And also a recent idea is that pressure is responsible for the signal given to cells either to divide and continue to grow or to die. And just to show that this is a domain of science which is progressing, I can say that 20 years ago, people would have considered living tissue like that as more as an ecological point of view, saying that you have a notion of carrying capacity, giving you how many cells you can put. Now this is no longer really the case, and I will show you the Conte sequences in terms of models. So this is more or less what is a living tissue. Just to give exactly at the opposite, what people are doing with that in terms of simulations, and what I would like here to point out, and I will not go in this direction, but just to say that there are teams now, there are companies now which are using mathematics to predict on real images, and what you see on the top here are real images taken by a slice of a 3D patient that try to predict what will be the progression of a tumor in order to predict the best time for and the best treatment possible. So there are many teams who are doing that now in the world, and some of them are successful. So this is a subject which is becoming used by medical doctors. And of course, what I will do is mathematics. I will simplify a little bit, not a little bit, very much the modeling. But if you want to be successful in this kind of pictures, what you need to do is put much more physics or much more biology than what I will do. And I will simplify it, but all these models which are used are more or less the same type of equations, but including what you need to include when you treat a given tumor. All tumors are different, in different organs are different, and so you put the minimum biology you need to represent the phenomena. And all this is listed here, and of course it's only a partial listing. So in terms of models, because I told you that you can consider a fluid tumor, a tissue, living tissue, like as a multi-phase flow and a multi-phase field, you are one of the most common models which are used are Canillard equations. These are degenerate and singular Canillard equations which are adapted to the living tissues, which are a little bit different from those which are used in material science, but not so much, a little bit more nonlinear. So there is an activity on that. I will not speak about that. I will speak of simpler models which are in between parabolic and hyperbolic. Canillard are fourth order. I will stay with second order equations, ignoring, if you want, the surface tension. What I show you here are pictures by the group in Milano where they compute with Canillard equations. This is where you see how these phase transitions can occur, which is something you find in experiments. It's not that it's not an image of theoreticians. It's what an observation of experimentalists. And here I show you on the right computations by a colleague of mine, Pierre Richard, which uses not exactly Canillard equations, but aggregation equations, non-local equations. And depending on the interactions of the two types of cells she puts, what you see is that either you can get an homogeneous tissues where the two types of cells want to live together, or they want to separate. And that depends on the interaction. They want to separate and create boundaries. And this is exactly what you observe in tissues. In some cases, the cells are very well separated. In some cases, the cells are well homogenized. To give you an example of a contact we had with immunotherapy of cancer, one of the reasons when immunotherapy fails is when the immune cells will go in the situation of a front, and they will not mix homogeneously, and then they are not efficient to attack the cancer cells, and the immunotherapy will not work. In the other case, it will work. So this is an example where you see the importance of this kind of questions. Of course, there are other questions. So this is what are called phase separation or segregation. Of course, existence regularity for this equation is also important. These are nonlinear models, which are not always very well understood. And of course, what you would like also to understand are asymptotic analysis, relations between different levels of modeling that you find in this area. So just to give you the general point of view of what I will explain in this talk, I would tell you that you can divide models in two types. There are what I call the compressible models, where you suppose that you have a pressure law, which is well known. So you assume that the pressure that I was speaking of about the tissue is a function of the cell density. So I call N1, N2, and maybe more, but I will stop to 2, it's enough for mathematics, cell types. I will have a pressure. And because of this extracellular matrix, it's like a porous media, so you see that the velocity is minus the gradient of a pressure. So the equations are the cells that are only moving. That's an assumption that they are only moving by a passive movement. The pressure, the cell division pushes the other cells away. And so the cells are moving because they are pushed away by the other cells. So this is the divergence of NV. And then you have a growth term, because you are living tissue, you have a growth term, and maybe an exchange term, which makes that cells can change type. For example, quiescent cells can become proliferative, or proliferative cells can become quiescent. So you can have interaction between the type of cells. And so this is the type of equation we'll find in the compressible case. What I would like to say and to insist of that now we consider that the growth of a tissue is mostly controlled by the pressure. And this has a name, it's contact inhibitions. Cells have ways to react chemically to a mechanical signal. So if cells are close each other, then they can bind, and this binding includes a chemical response inside the cell, which will tell to the cells, stop dividing, the pressure is too high, or you can divide, there is place, the pressure is low. So this has been, I think, the first persons who proposed this point of view are Bern and Drasdo, and there were physicists in France and Germany who made experiments showing that indeed you can play on the pressure to increase or decrease the cell division rate. And these people came with a notion of homeostatic pressure, which is the pressure after which the cells will begin to die. So there is a maximum pressure for the cell division. Afterwards, they will go to apoptosis. So the weakness of this model is that nobody is able so far to decide what is a pressure law, the law of state in a living tissue. Nobody can measure a pressure precisely enough to decide what it is. So in this talk, I will take always an homogeneous pressure just as a model. Nobody knows exactly what should be the pressure law. And on the other hand, you have incompressible models. So incompressible models are more or less the same equations. You don't change the equations. You always say that you have a Darcy law, but you add an incompressibility condition, n equal n1 plus n2. The total number of cells is 1. And if you do that, you add up the two equations, you get that the divergence of V is minus Laplace P, and this is given by the right-hand side. where here F is F1 plus F2, G equal G1 plus G2. And this gives you an equation. You solve it to find the pressure. When you know the pressure, you can go back to the equations and compute. So these models are interesting because mathematically, at least, it is not true that if you take an initial condition, N1 plus N2 will always be equal to 1. It might be that it's not true initially, for example. And so there will be a set where, so omega t should be the set where n of xt equal 1, and there is a set where n of xt is not equal to 1. And though this argument, writing this equation by summing, is only true in the saturated phase, but there is an insaturated phase and there is a free boundary. And this is an object of interest. And especially, we would like to understand the connection between this incompressible point of view and the compressible point of view. So what I will do in this talk, I will first explain what happens with a single equation. I will show you why Aronson-Benyon estimates are important to understand this equation. And then I will go to the multi-species and the incompressible limit. So in a single equation, you say you have a single type of cells. So you write a divergence of dTn plus divergence NV equal Ng of P, V minus grad P. So you recover the porous medium equation with the right-hand side. The right-hand side is nice because it is made in such a way, sorry about that, it is made in such a way that if the pressure is too high, is reaching this homeostatic pressure, then the right-hand side vanishes. And so you are in a very good situation, which is very, very close to the post-medium equation. More or less mathematics is known since a long time. And what we know is that the cell density is non-negative. The pressure is controlled by the homeostatic pressure if it is true initially, which we will assume. You have a control in L1, you have a control in BV. All the derivatives in L1 are bounded or at least exponentially growing with maximum growth given by the right-hand side. And we know that dTn is positive implies that dTn is positive initially implies that it's true for every time, which is a way to create a BV estimate in time. And this is extremely robust. What will be interesting in the future, let me tell you already, is what happens when gamma is very large. So I will be interested in the limit gamma tends to infinity, and this limit, and all these estimates are uniform in gamma. They don't depend on gamma. They are uniform. This idea that that the N0 is specific to get this BV bound is not really useful. You have an estimate which is more general, which tells you that whatever you do initially, dTn will be always controlled from below by an expression which is this one. So you have a minus k over t, which makes that at t equals 0, this is minus infinity, which means that you don't know anything at t equals 0, which is what we want. And when t is increasing, basically the exponential term will make it decay to 0. So this model is, in some sense, extremely simplified, and a tumor is always growing. So it doesn't contain all the biology you would like to contain, but mathematically it's simple. It's also interesting to see that the exponential rate is a mixture between the gamma, which is the pressure law you put in the first order term, and a number which only come from the right-hand side, which means that by opposition to what one could think, the right-hand side is not only a perturbation of the first order, on the second order equation of the Paul's medium equations. There is a deep interaction between the two, which creates something new. And this sign is this product where you see the interaction between the two terms. This estimate is due to Aronson and Benyon in the 80s, end of the 70s. Okay, the mathematics is very simple, but numerics tells you that there is something interesting. So here is what you observe on an example of two tumors which would be initiated at the different points and which would be growing independently. So you see that so what you see in black is the number of cells. What you see in blue is the pressure. So you see that the pressure begins to increase only when n is large enough, the number of cells is large enough. Then you have three boundaries which are created. These three boundaries are moving while the number of cells also increases. And there is an interaction which makes at some points they join and they create discontinuities. In fact, it's a numerical, theoretically, it's a discontinuity. The numerics is not very good. And you have some discontinuities where you join, or where these new islands, these islands or these tumors join and create new objects. Of course, it's a mathematical question, but this is showing that there is something complicated which is behind these equations that you would like to understand. So I will tell you first about the Aronson-Benillan estimate that I told you is important. So Aronson-Benillan estimate is an estimate coming from porous medium equation. If you ignore the right-hand side, you multiply the equation by n gamma minus 1 to get an equation of the pressure. It turns out that the equation of the pressure is self-contained and looks like an Hamilton-Jacobi equation. If you compute the Laplacian of the pressure, you call it W, you differentiate twice the equation on P, what you find is an operator in the strong form, advection, a positive term, another advection term, and a term which is an elliptic term in a strong form, and then a term which is gamma W squared, which makes as the solution, the regularity will stop, the solution, the W will blow up because dTW equal W squared will blow up, which is the reason why the solution is not very smooth. But you have Baron-Blatt solutions with corners, where the solution is not extremely smooth. So the observation of Benillan and Aronson is that you have a solution, dTW, because this term, positive term, is a super solution of this equation, and you have a solution which is minus 1 over gamma t, which is a solution of it. So W is always larger than minus 1 over gamma t, because this is a super solution is larger than a solution, especially because at time t equals 0, it is true. This one is minus infinity. So this is the idea of Aronson-Benillan. If you want to make it work with the right-hand side, what you discover is that the term which is of interest is no longer Laplace P. It's the combination of Laplace P plus G. This is the term which comes with a gamma. And if you want something which is uniform in gamma, the only hope is that you can estimate this term. And it's what happens if you compute the equation on this W here. What you find is an operator in strong form, a term in gamma W squared as before, and you see that now you generate a term which is a positive term. On the right hand side, it's a positive term. It's an absorption term. It's an absorption term. So it's a negative term. Sorry. So if you put here, it's a negative term, which makes that this is an absorption term, which is responsible an exponential decay. So in some sense, because of this interaction, the term G of P which is a growth term, is also a good term in terms of estimating the Laplace, the pressure or the time derivative of P. It's an absorption term which have an exponential damping. So this is what is behind all these estimates. So this is for a single equation, and you understand the single equation satisfies the comparison principle. It plays an important role. If you do now a system where you put the same velocity in the two equations, because the cells behave in the same way in terms of the pressure, when you add up, you seem to be in the good shape, because more or less you get always a porous medium equation with the right-hand side. Except now things are much more complicated, and all the arguments we had before fail completely. So for example, I told you that it is BV. This is not true, except in 1D. In 1D, you can still find BV bounds because of the miracle of algebra in 1D. In higher dimensions, there is no BV bounds. If you look at the pressure equation, I told you it's nice because the pressure equation is self-contained. It's no longer true. The right-hand side contains N1, N2, and P. So if you want to do, for example, an Aronson-Benyon estimate, where you have to differentiate twice, you are facing a big problem because you should differentiate the right-hand side also, because I told you that the good term in the Aronson-Benyon estimate contains the growth term, and this one contains N1 and N2, which are even not BV. So it's complicated. And the outcome, of course, is that if you want to do something like existence or stability, of course, what you want to know is compactness in this term N grad P. So you need compactness, so you need to find it somewhere, either by BV bounds or by Aronson-Benyon or somewhere. And this is a difficulty. So in terms of existence for this problem, the situation is much more open than for a single equation. So the situation is the following. If you assume positivity, N1 plus N2 strictly positive, then the equation, which I was mentioning here, becomes parabolic, non-degenerate. So you have regularizing effect. You can use regularizing effects, and then you have you can go further and prove that there are solutions that are smooth and so on. You have tools to do that. These are works by Michel Birch, Daniel Hilortz, and later Mayan Mimura. If you want to also handle the case where you have N1 plus N2, possibly 0, that's what's called our attention. There is a paper by Carrillo, Sant'Ambrogio, Marcus Schmidtchen, and co-authors, which in dimension 1 use the BV estimate on N1 on the ratio N1 over N, because it satisfies an equation in a strong form, and they prove BV estimates, and they got existence like that. So what we decided is to work on this with the Polisher team. We managed to use the Aronson-Benyon estimates in some way and got existence with not only existence, but also second order estimates under the condition that f of 0 is g of 0. Recently, there is a paper by Price and Xu and another one with Jianguo Liu, where they use only compactness. So they don't need this condition f of 0, g of 0 are equal. It's a simplification of the assumptions, better result. So this is the status of existence now for this system. So we don't know, for example, if the solutions have a second derivative bounded, like in the Barat-Bellat case, when f of 0 is not equal to g of 0. It's an open question. So now let me tell you about incompressible limits and the free boundary problem. So the free boundary problem for tumor growth is to say that your tumor is in the set omega of t, that the boundary is moving with a certain velocity, which is given by Darcy law, and that the pressure is given by minus Laplace p equal g of p, with a boundary which is with a Dirichlet boundary condition. So this is a geometric point of view of the incompressible flow. In reality, people prefer to put a pressure which on the boundary is proportional to the curvature, but I've never seen a derivation of such a rigorous derivation of such a law as I will do now when p equals 0 on the boundary. So the idea to connect the compressible porous medium equation with this free boundary problem is something which we call the LHO. I mean, this problem is called the LHO problem, by the way. So what we call the LHO limit, which is a stiff pressure law limit, which means that gamma equal to infinity. So when gamma tends to infinity, the pressure will become extremely big, I mean, growing quickly when n is larger than 1. And so this imposes that n is less than 1. So it's an old problem which was studied in the 70s, which was derived as gamma tends to infinity by semi-group methods by Benilan, Bida, and for which the Spanish team has been very active, and the team of Caffarelli, Salsa, and others have been very active on proving regularity of the free boundary. So it's a different problem. So recently, people have been working again on that with different methods. So there is a method by Inwon Kim and Antoine Mellet, which was a method used by Caffarelli, which is to see an obstacle problem behind all this. So in this, there is this bio-key change of variable, which makes that there is an obstacle problem occurring somewhere. There is a viscosity solution point of view. There is an Hamilton-Jacobi equation, so it's natural to use viscosity solution. That's what Inwon Kim did. There is a more recent tool, which is optimal transportation, which has been very much used by Sant'Ambrogio and Mori and their students. This, of course, is when G equals 0, because these are only restricted to the conservative case. And they call it congestion. What I call incompressible limit, they call it congestions. It's not the same application they have in mind. So let me tell you what happens. When gamma tends to infinity, what we know is that n gamma will converge strongly, the pressure will converge strongly. They are controlled from above, both of them. The gradient of the pressure, you have bounded it, so it's converged weakly in L2. And so because of strong weak convergence, you can pass the limit, no problem. You find that the equation is true in the limit. The only thing which is weak is a relation n to the power gamma, which tells you that if n infinity is strictly less than 1, p equals 0, and nothing else. You don't know more than that. And what is a miracle is that this problem with this relation very weak between n infinity and p infinity has a unique weak solution. And there are several proofs of that. There is a proof a la Olenik, which we did with Vasquez and Quiros. There is a recent proof by entropy by Bida. There is also another proof in Sobolev spaces, which we are working on. So in some sense, you have a weak limit, which is a weak version of the LHO problem, of the weak formulation of the LHO problem. So this is easy to prove. The real problem to prove is that the complementarity condition relation is true. That you can argue that either p equals 0, and then this product here is 0, or if p is not 0, n equal 1, and if n equal 1, dt n equal 1, and you write that Laplace p equals g of p. So you would like to argue that there are two zones, a zone where n equal 1 and Laplace p plus g of p equals 0, and a zone where n is less than 1 and p equals 0. So you would like to establish this, but it's not easy, completely easy, because more or less to establish this is the same as to establish that the gradient p converges strongly and not weakly to p infinity. And this is the real complicated theorem. So to do that, the proof is to use a pressure equation to notice that the term gamma is in front of exactly this complementary condition. So in principle, formally, it's easy to say that this will be 0 in the limit, except you have problems in strong compactness of the gradient p squared. That's the reason why to prove this limit, you need a strong compactness of the gradient, and so you need Aronson-Benilan. Aronson-Benilan is telling you that in X you have a control of the Laplacian in one direction, but that's enough. And if you control the Laplacian, then the grad p is compact in X. So and the time derivative is also a problem, and this uses, there is an argument which uses dTp is greater of 0. So this is the way it works, a little bit complicated and technical to establish all this, but it works. All this is not stupid, because if you look at experiments, what you see on in vitro, if you have a dish where you grow cancer cells, here's the center of the dish, what you see is that the number of cells is more or less constant up to statistical errors, and then you have a clear free boundary. And here's the same with different nutrients concentrations, and what you see is more or less a free boundary, the cell density is constant, and then you see that cells are dying in the middle because they don't have food, and food is not included in this model. These are other examples of this growing culture of cells. For systems, if you want to prove that the complementarity condition is true for systems, is much more complicated. So far, the only result we could do is in dimension 1. And we cannot prove more than, and we use BV estimates very strongly. Dimension 1 is used at several points here. So it's difficult to go further. It's more or less an open question to prove this, this gamma tends to infinity limit when you are in multi-game. To conclude, I would like to speak of this problem of the nutrients and the necrotic core. So if you want to put nutrients, what you do is you add up an equation saying that the nutrients, which concentration is C, is diffused. So it's always a diffusion equation. You have absorption of the nutrient by the cells, and you have release by, let's say, blood vessels. So you have an equation like that, and you say that the growth is controlled both the pressure and the nutrients. The problem is that this system is not a good system. It's not a cooperative system. It's not a competitive system. So it fails exactly in the bad systems where you don't have a comparison principle. And so most of the tools we have been using so far cancel. And so it was a little bit complicated to study the elimination limit, to establish that the limiting equation is true, that the weak formulation is true, and uniqueness is not a problem. These are the easy part. What is difficult is to establish the complementarity condition. And that was a long-term open problem that we solved recently with a PhD student, Noemi David from Bologna. And what we found that we have to do for that is first to change the Aronson-Benillan estimate, which is a control from in L infinity from below, by an L2 estimate, which is easy, which is weaker, but which is easier to apply to a system like that, because we don't require all the regularity of an L infinity estimate. And what we proved, and we are very proud of that, is that the gradient of P is in L4 uniformly in gamma. That in the porous medium equation, it's already true for the porous medium equation, you have better than L2, you have L4, and this is uniform in gamma, and this is sharp. You cannot improve the 4 here. And because of that, though we have compactness in space coming from there, and this gives you what is missing for the limiting problem, which, because now we don't have DTP. The problem is that we don't have DTP is positive in this case. Because of nutrients, cells can die. This is what you see on this computation. Now, if you see this, okay, I have to stop now. So this is what you see, what happens here. You have a tumor growing. You have, so at the beginning, it's growing like a traveling wave, a spherical wave. But after some time, the nutrient, which is released from the outside in this computation, doesn't reach the center of the cells. The nutrients are eaten by the cells which are dividing. So in the middle, cells are beginning to die. And if you go further, the cells are still continuing to die. And the middle of the cell, the tumor becomes completely necrotic. So what you recover is that you have a tumor is made of a proliferating cells on the boundary, a necrotic core. This is what you observe in the picture I showed at the beginning. And associated to that, you can have also in some cases, interface instability. And so the tumor can begin to lose its sphericality and becomes unstable and create fingering. This is related, of course, to something which is well known in thermochemical reaction, where the system is much simpler. It's made of two heat equations coupled by this bad coupling, which is where comparison principle is wrong. And it is known that this one creates something which we can call a dynamical Turing instability, when alpha has to be small enough. And what you observe in this chemical reaction is that the spherical wave that is initially true is destabilized and creates this kind of frame front, which is unstable. So to conclude this talk, what I wanted to show is that the modeling of living tissues of tumor growth is a very active scientific field, not only in mathematics, also in mechanics and in physics, with new ideas coming out. That asymptotic analysis and multiscale methods are very present because of different scales, so from molecules to cells to organs. You have many scales in these biology problems. And one of the difficulties is always that because of the complexity of biology, you always have to face systems which are I would say bad systems. If a good system can exist, so I don't know, but these are bad systems for sure, and they create instability, which is one of which is a sign of the difficulties of the analysis. All this has been done with all these people, and I will stop here, keeping their face on the screen.
Thank you very much for the very interesting and nice talk. We have time for discussion, questions, comments from the audience. Please, if you have questions, switch on the microphone.
I have a question.
Please, Tommaso.
Benoît, in all this kind of, I have two questions in reality. One is mathematical. In all of this kind of model, you take a Darcy law, and therefore substantially you have a parabolic structure. If you take a reality momentum equation, also if it's small, maybe the inertial term, you have a parabolic model, and therefore regularity may be completely different. Therefore, what do you have a comment about this? Second question I put after.
So for this question, I will tell you, the Darcy law is very realistic. So it's many people are happy with this, but not always, because there are experiments which are not compatible with that. So there have been several papers, several extensions with keeping. So this comes from the Navier-Stokes equation. So you can keep also the viscosity, and then, as you mentioned, things are much more complicated. Adding this viscosity is not always a simplification in terms of mathematics, but there have been several papers on that. So extensions, basically to recover the incompressible limit.
My second question is in some sense a little provocative. Usually this model that arrive from physics, and then you adapt in this biological framework. But in physics, you have some general principle. For example, to fix production, you have like entropy principle or the things Galilean invariance, many general rule. Here you don't have any help in some sense. Now, for example, you put this FG in the production term, and then you give a beautiful figure. But FG, how you, in practical purpose, you fix? In which manner, for example, also the assumption depend on pressure. How is justified this?
Okay, of course, this is the problem of the modeling. So we decide at some point that we try this to explain it with pressure, for example, not with nutrient, and we see the consequences. And it turns out that the consequences are compatible with the observations. Nevertheless, the fact that the growth is controlled by pressure, this was bigger at the beginning, was just a numerical evidence by Bird and Drasdo. They computing and they said no, it can, if you want to fit with the numbers, we have to put it like that. And then there were experiments, and those are experiments where they put living cells and they apply pressure. They apply pressure, and then they compute the division rates, because they have ways to visualize division rates okay, there are experimental devices to do that. And so they could really measure the pressures. With these homeostatic pressures, they can measure it. So it's not only a vision of mathematicians, it's also something you can measure experimentally.
Yes. Yes, of course, we have so many degrees of freedom. How you not so sure if what you compare is.
Of course, all these are experiments in lab. Oh yes, but there is another picture I showed at the beginning, which is interesting. It's in vivo. It's a group of also in mechanics. They were applying pressure on the organ of a rat, and they measure what the effect is, and they saw what are the chemical reactions, I mean the enzymatic reactions which are produced by this pressure. And then they could completely understand the chemical cycle which has been produced by that, and explain observations. And this was related to cancer molecules, proteins which are typical of cancer, and how they are induced by pressure.
Okay,
so of course, we are not at the stage of physics where you can reproduce the experiments everywhere in the world and compute with 3 digits the outcome. It's not at that stage, but these are experiments that people can do in different labs, and they can
May I ask a question?
get consequences.
Yes, please, Marco.
I raised a hand. So the first question is that is there any estimate on the thickness of the interface with gamma? So how the interface shrinks when the gamma goes, or do you think this gamma sending it to infinity is just a mathematical artifact? And
we have error estimates. We know error estimates on the convergence rate. That is available. There are very few papers, but in one team has one, for example, in Wasserstein distance. But there is, yes, in fact, there is no really interface in that case. It's only in the limit that you have an interface.
Yeah, when gamma is infinite, but
if you're really now I'm not sure if you can give a. It's not like in Canillard. In Canillard, you can really have some kind of traveling wave. You can measure it in terms of the parameter in Canillard. But in porous medium equation, I'm not sure you have a way to make this statement rigorous.
And another question is that if I well understood, so at a certain stage, you had just that the gradient were converging weakly. Then using the Aronson-Benillan estimate, you were able to make it strong, right? But I'm sure there are conditions which maybe this Aronson-Benillan you cannot, it doesn't work because of the nutrients as a particularly difficult to treat form. So if you have this, you don't have the strong convergence, you might have phenomena like concentration or wide oscillations, which maybe are realistic because it's this oscillation of the interfaces sometimes are actually seen. So So, for example, in Rayleigh Taylor instability, that's what you have. You have this kind of wide oscillations of the in the limit of regularization.
Yes, you are correct. I think this, for example, if you put surface tension, which I didn't put in these models, I'm not sure what happens.
So this lack of convergence could mean something physically.
Yes, it might be that you are correct, and that the real model, if you include surface tension, doesn't have a strong convergence. That's a good question. It might be possible that it's more complicated than. I don't know any derivation of this surface tension in the limit, so it might be that it's connected to what you say.
Yes, also the gradient, the pressure are force. Some maybe you might in structural mechanics there are cases in which force are concentrated like deltas. So that
absolutely, yes. It's really meaningful. Yes, it could be that there is because you have in some sense a jump in the limit, so you should see it like that. Yes, yes, you are correct.
Thank you. Other questions, comments?
Just another curiosity. I mean, these models have been derived by microscopic models, more microscopic models.
There are these models by mostly in UK. They like to make a finer scale where they have what they call aggregation equations, non-local equations to see the interaction, the long-range interaction of cells. So this is a way to go to a scale which is a little bit smaller, and then you can derive it. It's yes, it's the non-local. You can recover the parabolic terms. You see, I'm not sure it has been done, but you can imagine it's possible to do it. Yes. But there is nothing like Boltzmann equations behind, because it's tissue is never rarified. It's not a good point of view here.
Okay.
More like an agent-based model or something.
Yes, absolutely, yes.
Other people want to ask something, comment something? Otherwise, we thank very much Professor Benoît Perthame.
So thank you very much for the invitation.
Thank you for your very nice talk and very clear. Maybe we can have 5 minutes break and we can resume at 4:30 with the second talk.
Sure, yeah, 4:30, right. Perfect.
Okay, see you then.
See you then.
Se vuoi intanto provare a condividere, così vediamo se funziona prima di ripartire.
Dovrebbe vedersi.
Vediamo? Ok, sì, poi metti pure a schermo intero. Sì, perfettamente.
Perfetto. Lo lascio così?
Va bene. Aspettiamo ancora un minuto, poi partiamo. Sì. It's a pleasure to have as a speaker Martina Conte, now at the University of Granada, but previously she has been a student of Parma University during her bachelor and master thesis. Then for the PhD, she moved to Spain, and now she is spending a postdoc period in Granada. She will speak about a multiscale approach to the study of brain tumor invasion and progression. Please.
Thank you. And good afternoon to everybody. First of all, I really would like to thank all the organizers for inviting me and giving me this opportunity. It's really a pleasure for me to be able to present you some of the results that I've been working on during my PhD. So today I would like to show you a couple of settings that we developed from a multiscale approach that has let's say, the final aim of analyzing specific mechanisms that are involved in the process of brain tumor invasion and progression. So just to give you an idea about the path I would like to follow for these seminars, we will briefly see the mathematical multiscale framework behind the different settings and from a general point of view, and then we are going to specify this setting in two different scenarios. So I'm going to present you two different models. They're actually, they are not, let's say, parallel works, they are kind of part of the same project. They are one step, they are one extension of the previous one. And the first work was done in collaboration with Professor Maria Gropri from the University of Parma and one of my two PhD supervisor, Professor Gerardo Giorda, that was in Beckham, where I did my PhD in Bilbao, and now is at the JKU in Austria. And we look specifically at the influence of the brain tissue structure on the progression of the tumor. And then, as a second step, I worked with Professor Surulescu from the University of Kaiserslautern in order to extend the model to take into account also the dynamics of the brain vasculatures and specific mechanisms that are driven by hypoxia-driven acidity. So for these two settings, we're going to see the main biological aspect and then the model setup and some of the numerical tests we performed. So starting from the general framework, the choice of using a multiscale approach comes from the fact that the biology that characterizes the tumor cell involves several phenomena, and they actually happen on different spatial and temporal scales. And this is why we choose a multiscale approach that in the specific is based on a kinetic formulation of the equation for the cell distribution. So in our settings, we're going to consider three levels of description. We start with a microscopic level at which we take into account that processes that actually happen at the level of a single cell. And for instance, we can describe the dynamics of specific receptors located on the cell membrane or the evolution of specific intracellular pathway involved in the general tumor progression. And this level is described using a variable y that is called the intermediate variable, whose dynamics are given by an ODE. So this equation actually, when the subcellular, the microscopic dynamics are much faster than the phenomena on the other level, that means the temporal scale of this microscopic level is much shorter, we consider a quasi-steady state approximation and we redefine this microscopic level, considering a new internal variable, there is nothing more than the deviation of y from its steady state. So to be a little bit more specific, in the two settings I'm going to show you today, at the microscopic level, we are going to model the dynamics of some membrane receptors that allow the cells to bind to the fibers and to crawl along the fibers. They are called integrins receptors. And the second family of transmembrane units, they are actually involved in the process of sensing of the extracellular pH level, and they are called proton sensing receptors. So this microscopic level is then included inside the mesoscopic one, at which what we described is the evolution of the cell distribution, considering the effect of different intercellular communication processes. So, for instance, we're gonna model the changes in the cell velocity that can originate from different external signals, or if we assume that there is a certain level of heterogeneity inside the tumor, we can model a phenotypic switch between different subpopulations of tumor cells. So the cell population is therefore described using a distribution function rho that depend on the time t, the position x, a microscopic velocity v, and an internal variable y. So the general kinetic transport equation has here on the left-hand side, the present or include the microscopic dynamics through the function g, while on the right-hand side, we have the turning operator L and E rho. There is an operators including all the conservative and non-conservative interaction between cell and microenvironment. In particular, L is the operators describing the cell velocity, and as I said, they can depend on different external signals. But in any case, it is described as a difference between a gain and a loose term, both involving a turning rate mu. They take into account, again, information from the microscopic level through the internal variable y, and then turning kernel t. There is the one describing the Q that is affecting the changes in the cell velocity. So this mesoscopic level is then upscaled, and to do this, we consider the 0 and first order moments of rho with respect to the internal variable z, the new internal variable, and then we integrate it again with respect to the macroscopic velocity. And from the mesoscopic equation, it's possible to derive the moments equation, whose spatial variable is rescaled with a small parameter epsilon. So in order to derive from this scaled equation, the equation at the macroscopic level, that is the one we actually simulate, what is possible to do is to use asymptotic methods that can rely on parabolic or hyperbolic limit, depending on the characteristic of the turning kernel or the relationship between the scales of the different phenomena. And in any case, we consider Hilbert expansion of the moments, and we equate the power of epsilon in this scaled equation to derive the macroscopic equation for capital M. In reality, what we derive is the equation. If we consider the Hilbert expansion of capital M, we derive the equation for the coefficient of the leading order of this expansion. And the macroscopic level equation basically allows us to observe or to describe that aspect of tumor evolution that can be also observed in the clinical context. So for instance, at this level, we can see the effect of cell proliferation or apoptosis, or the effect of different therapeutic treatment, or also looking at the healthy tissue, it is possible to look at the process of tissue degradation. So in this general framework, first, what we did was an analysis of a model with just one population involved, in which we look at how the structure of the brain tissue can influence the tumor progression. So here, actually, we look at a specific kind of brain tumor that is called glioma. And this is a malignant brain tumor that arises from the glia cell of the central nervous system. This cell in healthy situation provide nourishment and support to the neurons, but when a neoplasia arises, they actually start to become highly invasive and aggressive, and they acquired a strong migrating phenotype that determine that the typical infiltrative behaviors that we can observe from the clinical images of these brain tumors. So we look mainly at the process of cell migration, and at this stage, what we are interested in are the interaction between the tumor cell and, as I said, the nervous tissue. So the brain tissue is actually characterized by several tracts of fibers that basically connect different areas of the brain. This tract of alanine fibers for the cell represent kind of, let's say, highway leading and driving cell migration. So cell bind to the fibers and crawl along the fibers to migrate. And therefore, it is important to take into account information about this macroscopic structure of the brain tissue, because it highly influences the process of cell migration. And one way to take into account this kind of information is to consider DTI data. So this diffusion tensor imaging data are a medical imaging technique that basically allow us to quantify or to basically it gives us a measure of the apparent diffusivity of the water molecules in the brain. So what we are able to reconstruct processing DTI data is a water diffusion tensor. So for each voxel of the mesh in which the images of the brain can be divided, we can reconstruct a tensor that, of course, can be represented as an ellipsoid. And this ellipsoid is going to have a more spherical shape in that region where the tissue is more isotropic. That means there is no preferential direction for cell migration. Or it's going to have a more squeezed shape in one specific direction if the tissue is more anisotropic. Anisotropic means that there is actually in that specific voxel a path of aligned fibers that represent the preferential direction for cell migration. From the water diffusion tensor, it is possible then to derive the tensor related to tumor diffusion, and we're going to see one possible way in which this can be done, and it allows us to include at the macroscopic level the information that we get from the data. So going to the model, what we actually built is a one population model. So we describe only tumor cell evolution. At this stage, the internal variable Y represent the concentration of bounded integrins, so that membrane receptors that are already bounded to the fibers. And instead, the turning operators at the mesoscopic level is given by a turning rate lambda that depend on the microscopic variable, and a turning kernel that take into account information from the of the brain fibers through this function Q. So this function Q is called the fiber distribution function, and it is built starting from the information we got from the water diffusion tensor. And this is the way it's possible to describe the brain fibers as the dominant directional Q leading the changes in the cell velocity. So this turning operator is then included in the mesoscopic level equation. So we have our L operators on the right-hand side, coupled with an operator P describing the process of cell proliferation. And in this case, tumor cell proliferation is modeled as mediated by the presence of the healthy tissue. And finally, we have a loose term. So I didn't say it before, but we also included in the model a certain type of therapeutic treatment because we wanted to understand if it could affect somehow the interaction between the cell and the fibers. So we modeled a combination of radio and chemotherapy. The radiotherapy is included over here, and it is directly aimed at cell killing. And instead, the chemotherapy is modeling a drug that basically, some drugs that are integrins inhibitors. So this drug basically reduces or inhibits the binding affinity between the cell and the fibers, so reducing the capability of the cell to bind to the fibers and to move along them. And this is why we include this chemotherapeutic effect at the microscopic level, where we model the dynamics of the integrins. And here in the function G coming from this microscopic level, we have the dependency on DC. There is actually the chemotherapy dose. So this mesoscopic equation is then upscaled. At the macroscopic level, what we got is a diffusion advection reaction equation, where we see the presence of the tumor diffusion tensor built from the fiber distribution function involved in the turning operator. So this is the way DT is connected to the water diffusion tensor, and therefore to the information about the fiber structure coming from the DTI data. So this model was then numerically simulated. There was a first pre-processing step, because what we wanted were a realistic reconstruction of the brain geometry, and on this geometry, information about the fibers. So we processed MRI data in order to reconstruct the brain. We extracted 2D slice that is going to constitute our computational domain. We actually have the entire slice of both hemispheres. This is just a matter of visualization. And on this specific slice, we reconstruct the information about the DTI. So from the DTI, we were able to visualize a map of the anisotropy. So this map basically said that where we see a yellow and white area, these are area of high anisotropy, so area of strongly aligned fibers. While when we are in black or red area, these are area of low anisotropy, so basically isotropic tissue. If we zoom in one of these subregions and we plot the leading eigenvector of this the water diffusion tensor, we can somehow visualize or have an idea about the alignment of these fibers. So for instance, here there is clearly a path of fibers moving in the up-down direction. This information represents the basis for the simulation, and I'm going to show you a couple of numerical tests. The first one was done in order to understand which could be the response of the tumor to different kind of fiber distribution function. So in the literature, there are several possible expression for the function Q. And what we decided to do was to consider the three most used and compare how the tumor spread was responding to this different expression. So on the right-hand side here, you see the different tumor diffusion tensor that can be reconstructed from the different fiber distribution function. And as you can see, in the first case, the tumor diffusion tensor has an isotropic component proportional to the identity matrix that cannot be weighted in any way, and that somehow is including in the spreading of the tumor a certain level of isotropy that is not controlled, so it doesn't depend on the real tissue structure. On the other side, in the second case, this component can be actually weighted thanks to two parameters, delta and k. And in this case, if it is possible to have data that we can use to tune the model and to get a really precise estimation on k and delta, this distribution can be really powerful also because the computational cost is really low. For our case, we actually didn't have access to such data, and therefore we rely on the third distribution. There is something in the middle. So here we do not have any component, any isotropic component to weight. Everything depends just on the water diffusion tensor. And what we observe from the picture is that this distribution that is called ODF is also able to show us a spreading of the tumor in which there is the emergence of this branching or finger-like pattern in the evolution. There is a typical features that we also observe in the clinical images of the brain tumor. So for these reasons, we decided to rely on this distribution for the further analysis. And one of the other tests that we look at is the effect of the chemotherapy. So as I said, the chemotherapy is not killing cell. We consider a drug that inhibits just the integrins activity, so reduce the motility. And here what we plot is the difference between the solution of the model without therapy and the solution of the model when we apply chemotherapy. So the negative value, that means the blue region, referred to the region in which the model with the therapy show higher density. So if you think that the initial condition is located over here, we can see how the darkest blue region are closer to the initial condition. That means that when we apply therapy, actually cells seems to stay more close to the initial location, so less able to spread. And also they are less able to adapt to the microscopic changes in the structure. So when the tissue is changing pretty often, frequently, the model in which we apply chemotherapy seems to be not responding to these changes. So cells are following the main path of fibers, but they do not really interact that well with the tissue with respect to the case in which, of course, chemotherapy is not applied. So this model was the first step. Then we extended, and the aim was to understand, or to not to understand, but to model the interaction between tumor cell and the population of healthy cells that are called endothelial cell. These are the ones involved in the process of tumor angiogenesis. So angiogenesis refers to both the remodeling of existing vessel or the formation of new blood vessels from scratch. And tumor angiogenesis is promoted by the tumor cell when there are situations of let's say, limitation in the oxygen and nutrient supply. There is called as hypoxia, or when there are low pH conditions. This situation is called acidosis, and actually hypoxia and acidity are not independent, but they're strongly connected, because often what happens is that in hypoxic situation, tumor cells start to switch their metabolism toward a more glycolytic metabolism. That means they consume less oxygen, but they produce and release in the environment an acid that is called lactate, that basically is responsible for the microenvironmental acidosis. So we talk about hypoxia-driven acidity in the model. And concerning angiogenesis, we are mainly interested in two hallmarks that arise in the case of brain tumors. So endothelial cells, when they are attracted towards the tumors by specific growth factors produced by the tumor cell, they start to proliferate, showing a phenomena called microvascular hyperplasia that can be, for instance, observed over here in this histopathological sample, where we have this strong magenta island here in the middle of the tumors. These are complex structures called glomeruloid bodies that are basically made up of a mixture of strongly proliferating endothelial cell and muscle cell that are typical hallmark of angiogenesis in the highest grade glioma. And the second hallmark that has been observed in the case of tumor angiogenesis is the emergence in the tumor core of necrotic region, like for instance this clear region here in the middle. And these are the two main aspects we would like to capture with the settings that I'm gonna describe you right now. So with respect to the previous model, here we couple the dynamics of three different populations at the mesoscopic level. So we decided to include a certain level of intratumor heterogeneity, assuming that there is a subpopulation of tumor cell that just move without proliferates, and the second population that instead do not move but it proliferates. These populations are given by rho and R, and there can be a switch between moving and proliferating tumor cell when there are certain extracellular or microenvironmental conditions. And also in this case, the tumor population depend on a macroscopic variable Y. This idea of considering intratumor heterogeneity comes from the so-called Gorgo hypothesis. There is a biological hypothesis that basically refers to this fact that it seems that in the tumors there are these two subpopulations of tumor cell that evolve at the same time. The tumor population is then coupled with the dynamics of the endothelial cell described by W, and here we do not have any dependence on a microscopic level. So the internal variable for the tumor here represents the concentration of bonded integrin spheres and also occupied proton sensing receptors. While going to the mesoscopic level, the turning operator for tumor cell is exactly the same of the first model. So again, we have the brain fibers as dominant directional cue for the tumor cell. And instead, for the endothelial cell orientation, what we model is a response of the endothelial cells towards gradient of proliferating tumor cell. So what happened in biology is that tumor cells produce specific growth factors that are released in the environment, and the gradient of these factors determine a chemotatic force for the endothelial cell orientation. Since we do not want to include, at least at this stage, a frightened variable also modeling the dynamics of these growth factors, we decided to directly connect the orientation of the endothelial cells to the density of that cells that actually produce these growth factors. So in the turning rate eta, we have the dependency on capital R, and also it is described as a turning frequency eta 0 multiplied by the exponent of the pathway gradient of R multiplied by a function. This function AR is the one taking into account, for instance, the equilibrium of the endothelial cell receptor binding. So to complete the mesoscopic level, we also include phenotypic switches between moving and proliferating tumor cell. And we model proliferation for both tumor and endothelial cell. For the tumor cell, proliferation is mediated by the presence of the healthy tissue and the availability of nutrients provided in this case by the availability of endothelial cell. While for the endothelial cell, we just have a tissue-mediated proliferation. A final operator that we actually also include is called acid-induced death term. What we wanted to model is the fact that even if tumor cell seems to be more resistant to low pH condition than healthy cell, there are certain level of pH under which also tumor cells start to die. And therefore, we want to include this threshold as a kind of apoptotic term for the tumor population. And this is how it looks like, the mesoscopic model. So we have two kinetic transport equations for moving glioma cell and endothelial cell. And we see, and instead for the resting population, we have an integral differential equation. We can see the two turning operators for tumor cell, L lambda, and for endothelial cell, L eta, coupled with the operator describing the phenotypic switch. So as I said, we have a switch from migrating towards proliferating tumor cell. There is described by this beta, when there is a pH condition in the extracellular space too low. So it means that when the extracellular space is too acid, also tumor cells start to move away. Capital S is a macroscopic variable that actually models the concentration of these protons in the microenvironment. Alpha and LM are instead operators referring to the switch from migrating towards proliferating tumor cell. Alpha model the fact that when there are nutrients available and there is an optimal pH condition, tumor cell actually stop and grow. And instead LM, it depends on capital N. There is the total macroscopic density of tumor cells, so the sum of migrating and proliferating cell. And this switching, when I model the fact that when the environment is too crowded, tumor cells cannot move anymore, they do not have space to move, but if there is still some space, they are allowed to proliferate. PR and PW are the operator describing proliferation of the cell, and finally gamma is the operator I told you before that includes this apoptotic term tumor cell. So the model is upscaled with a parabolic limit, and at the macroscopic level, we got a system of two equations, one for the total tumor population and one for the endothelial cell. So we have two diffusion advection reaction equations. In particular, for tumor cell, we see the presence of two functions, phi and bar rho, that depend on the fact that we have to couple two equations for migrating and proliferating cell to obtain this overall, let's say, equation. Bar rho somehow is a function limiting the motility coefficient. We have FQS, there actually is a function taking into account information from the microscopic level, and the transport term is driven by the gradient of Q, that is the fraction of healthy tissue available. So this model somehow an apoptotic force driving cell transport. And it is also driven by the gradient of the concentration of extracellular protons. And here with the minus in front is modeling a kind of pH repellent axis. The equation for the endothelial cell is instead described with a transport term that depend on the gradient of just one of the two populations of tumor cell, making a nonlinear coupling between the two equations. And what we did was to directly to couple this system with a Farad equation directly stated at the macroscopic level that describe the evolution of the extracellular acidity, basically. So we have S, there are our protons, they are diffusing in the space, and with the reaction term, we model their production by the tumor cell and a process of buffering of acidity thanks to the availability of endothelial cells, so vasculatures through the function W. So this model was discretized and then simulated on the we simulated the macroscopic limit. And we consider these initial conditions. So we are still on our brain slice, similar to the one I showed you before, with same data from about the fibers. We assume a central core of proliferating tumor cell initially, an outer rim of tumor cell, migrating cell. So when we couple them, we obtain these initial conditions for tumor, kind of really simple. The corresponding condition for the protons is translated here into level of pH. There is actually something the clinicians are able also to quantify. So there are techniques that allow them to observe and to quantify the different level of pH in the brain. And we couple it with initial condition for endothelial cell, really simplified. So we just consider three blood vessels located on the right-hand side of three conditions located on the right-hand side of the domain. They want to simulate kind of three blood vessels, not well developed yet. And I said it is really simplified because we know that in the brain there are much more vasculatures available, and therefore the fact that we just assume this three blood vessel is going to affect, of course, tumor proliferation because we are somehow limiting the nutrients they need. But our goal, at least at this stage, was to observe, to have a clear picture of the interaction between tumor and endothelial cell and to observe which kind of dynamics we got for endothelial cell. So one of the numerical results is simply the numerical simulation of the three population model. So we have our tumor cell divided into two subpopulations, the endothelial cell and the pH level. So tumor cells start to switch from proliferating towards migrating, since there is not enough nutrients for them to proliferate. As I said, it seems that there is a reduced spread, and it mainly depends on the fact that there is really a low level of nutrients for them. The pH level is decreasing because of the tumor activity. But the most interesting dynamics comes from the endothelial cells. So these cells are, of course, diffusing in the domain. They are growing, especially in this upper corner, because of the initial condition for the healthy tissue that there is below. But they also start to show a static effect towards the core of the tumor population, where initially the proliferating tumor cells were located. And when times pass, we obtain these results. So we obtain the emergence of this island of highly proliferating endothelial cell. There are actually a good representation of the phenomenon of microvascular hyperplasia that I was showing you before in the histopathological sample. So this island is highly dense island of strongly proliferating endothelial cell. There are one hallmark of angiogenesis for a brain tumor. This was the first result. A second analysis concerned instead the role of the phenotypic heterogeneity that we include in the settings. So as I said, we rely initially on this Gorg hypothesis that actually is still an under-debated questions in biology. And what we wanted to understand is which kind of effect, including or not this hypothesis, has on our setting. So we went back to the mesoscopic level. We redefined the model in order to exclude this hypothesis. So we have just one population. We deduce again the macroscopic level equation and we simulate them. Here we have the new variable, so the tumor population, and now we have just one tumor population, the endothelial cell and the pH level. So if we look at the results, qualitatively speaking, they're pretty similar to the previous one. Still, the tumor is spreading, is going towards the endothelial cell are moving towards the tumor. So we have this static force is stronger because now endothelial cell orientation is driven by the entire tumor mass. We see the decrease in the pH level, and we still captures the emergence of this microvascular hyperplasia. But the main difference between the previous result and this one seems to be connected to the velocity of the tumor progression. So it seems that forgetting about this level of phenotypic heterogeneity can give us a much faster progression of the tumors towards higher grade. And actually, this is something we observe better performing a further analysis, because we decided to extend the previous settings, including two further equations. So we coupled tumor endothelial cell and protons dynamics with an equation describing tissue degradation, healthy tissue degradation, and an equation describing the emergence of necrotic tissue. As I said, necrosis seems to be a hallmark of tumor angiogenesis. So we extend the model to this population model, and we define a possible grading for the tumor. So we define this function G that represents duration between the volume of the necrotic core over the overall tumor volume. So we decided to consider a necrosis-based tumor grading because there are references in the literature that says basically that the overall survival of a patient that has this kind of neoplasia, it seems to be more connected to the necrotic component of the tumor volume than to the tumor volume itself. So how big is the tumor? And therefore, we define this grading, and we observe how the percentage of necrosis can evolve over time in different scenarios. So we have three grades. So when we are below 25%, we talk about grade 1. When we are between 25 and 50%, we have second grades, and then we have the third grades over 30%. If we focus for a moment on the gray lines, the continuous one refer to the case, to the model in which we include the Gorg hypothesis. So we have the split of the tumor population. Instead, the dashed one refer to a model in which we forget about this Gorg hypothesis. So as you can see, the two lines is basically the same trend. We have just a slightly faster progression, for instance, from grade 2 to grade 3, that happens, let's say, slightly earlier in the case in which we do not have the Gorg hypothesis. And this is in line with the qualitative results. So as I told you before, just from the previous picture, it was clear that there was a slightly faster spread of the tumor, slightly faster progression. And this was the main effect, at least on our settings, of this intratumor heterogeneity. And this is confirmed also by this analysis. If you look instead at the two continuous lines, the gray one and the red one, the gray one is still the model with the Gorg hypothesis. Instead, the red one refers to a slightly modified model in which we do not include endothelial cell. So we just model tumor evolution without including all the aspects concerning or connected to angiogenesis. So here what we have is an initial window in which basically the progression of the tumor, the gray line, in which we have endothelial cell, seems to be slightly faster than the other case. So we go from grade 1 to grade 2 slightly earlier, let's say a couple of weeks before. But then the behavior is reversed. And in the case in which we do not have endothelial cell, the red line, we have just a much faster progression towards the highest grade with respect to the case in which we include these dynamics. It can seem paradoxical, but there is actually an explanation for these different behaviors. So initially what happened is that the endothelial cell that we include are not able to form a well-developed vasculatures that can buffer away acidity and therefore reduce a bit the necrosis. But at the same time, they are still able to support a bit proliferation. So this is why we have a slightly faster progression. But then the behavior is reversed because this endothelial cell created vasculatures that actually is buffering away acidity. So there is still an increase in the grades, but it's much slower respect to this case in which there is anything buffering away this acid, and therefore the percentage of necrosis just increase. So let me wrap up a bit these two settings and see which are the main perspective. So these are two steps of a more, let's say, bigger project with which we would like to analyze several mechanisms involved in the process of progression of these brain tumors. They are really complex to study also from a medical point of view. They often are a really poor prognosis for the patient. So looking at the perspective, what we are looking at right now is first a possible validation of the model prediction. So both for the results about the tumor response to the tissue anisotropy, that for the results about the hypoxia-related features, we would like to compare the model outcomes with real data. So for the first case would be simpler, but for the second one, before this comparison, it is needed to define a more realistic initial condition for endothelial cell. So something like analyzing vascular perfusion imaging data would be the way in which we can reconstruct a more realistic condition for this endothelial cell that would allow us to replicate more realistic also spreading of the tumor mass. A second point we are looking at is, as I tell you, we include here just the role of these growth factors in an indirect way. So we are looking right now at the way to include them directly and to include their influence on tumor dynamics. This project, this model has a different aim. What we would like to do here is to use an extension of our settings to evaluate the treatment efficacy of different combined therapies that are actually mainly aimed at affecting the process of tumor angiogenesis. And this model is going to have a deterministic part coupled with a stochastic one. They want to take into account the uncertainty that characterizes the treatment of this tumor in the clinical context. And finally, a third direction I would like to look at, and I'm trying to look at, is to change the healthy population of cells we include in the model and look instead at the interaction between tumor and immune cells, so cell of the immune system, in order to understand how and if the interaction between these different populations can lead to specific different infiltrative behavior of the tumor cell, and also with a final goal of looking at the possibility of defining optimal therapeutic approaches that can be based on a boost of the immune system from the outside. And that was more or less what I wanted to tell you. And so thank you very much to all of you for your attention. So let me leave the previous slide.
Thank you very much, Martina, for the very interesting talk. We have time for questions, comments, and for discussion. Andrea Tosin, please.
Thank you. Thank you, Martina, for your seminar. I just have two questions about, one mainly about the first part. When you introduced the internal variable Y or Z, if you look at perturbations with respect to the steady state, if I'm not wrong, you said something like the fact that the dynamics of this internal variable are quick.
Yes.
And my question now is twofold. On one hand, I would like to know quick with respect to what? So I would, I mean, with respect to which other time scale? And the second question is, if they are quick microscopic dynamics, why don't you have in your kinetic equation the kind of small or large parameter in front of the term of the divergence with respect to Z, which accounts for the fact that you are in a regime in which that microscopic process is quicker than, for instance, the transport in space or something like that.
Okay, so quick, when I said quick, I mean the macroscopic time scale is less than seconds. So it's as a temporal scale, there is microsecond or something similar. So I say quicker with respect to the temporal scale of the processes of proliferation, for instance, of tumor cell proliferation, of tumor migration. There is at least for what we were looking at is something of the order of at least hours or days. So the simulation were on 80 days, if I remember correctly. So this is why we consider just the deviation. So we can assume that macroscopic dynamics are always pretty close to the steady state when we look at this really larger temporal scale. And for the second question, I didn't actually look at it deeply. It's something I actually didn't think before. I think that just the assumption that we made on the macroscopic level, and then when we rescale the equation, so when we define the moment and we rescale the equation, temporally speaking, we included that stage, the parameters in front that take into account the fact that the dynamics are much faster. So we do not.
Okay, so this quicker time scale, it can be appreciated already at the macroscopic, but not at the mesoscopic level. So at the mesoscopic level, they are of the same order of magnitudes of other phenomena like the transport or other phenomena that you include on the right-hand side, right?
I think that they are faster, actually. Now I'm thinking about it. So they could be faster, they could be at same order, or actually faster. I would like to look deeply in it. I mean, I think that it's a really good point. I didn't think about it before. I just thought that the quantity-de-Secre approximation was enough to already considering this different scale, but perhaps it would be interesting to look at it from this point of view.
Okay.
Thank you.
And the other question was the numerical simulation you showed us were performed by discretizing directly the kinetic equation or the macroscopic limits? Of
I discretized the macroscopic limits, so the simulation of the macroscopic densities.
Thank you.
Okay, other questions, comments?
Can I ask, just something which caught my attention was when you were mentioning the switching between cells that were resident and the one so proliferating probably, and the other which were moving. So the process does make sense to think of the process as being a fast process. So the switching, I mean, faster because that's
to the macroscopic dynamics, or faster refer to which time scale?
Well, to the other time scale. I mean,
I think that
to the macroscopic dynamics. Well, you're just answer the question like this one, but because if usually when you have this kind of switching and it is fast with respect to other phenomena, you could derive, you could lower the dimensionality of your problem at the price of getting a nonlinear, some kind of nonlinear process, like nonlinear diffusion or something like that. So that's why I was asking. I mean, if you could derive an equation for just one species, so you could glue the resident, the proliferating and the moving cells in just one equation, in which the effect of the switching would be some kind of a nonlinear diffusion or some other kind of effect. So the process of the switching, is it fast or is it comparable to the rest?
It is for sure slower than, for instance, proliferation. So proliferation is the time scale of at least hours or a couple of days, depending on the stage of the tumor. So for sure it's faster, but I think it is comparable, for instance, with migration, so the transport term.
No, I mean the process of switching. So they switch fast. So they switch fast.
They switch fast, but not that fast like the macroscopic time scale. So it's exactly the one in the middle. It's much lower with respect to proliferation, for sure. It also, it also depends on the macroenvironmental condition. So how fast is the switch depend also on the macroenvironmental condition that leads this switching. So how fast we have the variation, for instance, of the pH level. Is that also a way?
So what maybe you said, I mean, I'm sure you said that, what causes the switching?
So basically, the cell sends the pH level. So when the environment is too acid, they migrate away. They want to move away. And instead, when there are nutrients available and a pretty good pH level, they go on the other side, or they stop and proliferate. And there was just an additional one for the too crowded environment. So when the environment was too crowded, they just do not have space to move. And they just to stop, and in that case, proliferate.
I see. Okay, so that maybe is something worth to look at. So, but the whole context was the Warburg effect. So the acidity was caused by the tumor cells.
Yes.
Okay, other comments, remarks? So if there are no other comments or questions, we thank very much both speakers of today. Very nice talk. Thanks, many thanks to all the participants, and see you next week. If other colleagues or students are interested in this series of seminars, please tell them to register using the Google form, and they will be included in the mailing list. So they could register also next week. There is time to add people, of course, to mailing list. Next week, the talks will be again on Friday afternoon. They will start at 3 p.m., not at 3:30 as today, but at 3 p.m. And we will have talks by Professor Jonathan Potts and by Rossella Della Marca, but you will receive the link and the reminder in due time. For now, that's all. Good afternoon to everybody, and see you next time.
Thank you. Thank you. Bye.
Thank you.
Bye.
My computer just decided I can't stop the.
Bye.